Good afternoon, everyone. Welcome back to FirstThought Focus. FirstThought Focus is a life sciences focused expert network with a mission to bring back subject matter expertise into the expert network industry. FirstThought Focus is our virtual conference series that aims to showcase the breadth and depth of FirstThought's experts while also covering a wide range of topics that are relevant and important to our industry. Last month, we wrapped up a 4-part series on CAR T and cell therapies. You can find the replays on our website, firstthought.io/focus. And today we are bringing you the second half of our series on innovations in research concerning the human microbiome. You can download the replay of part 1, which was hosted with Dr. Jen Wargo from MD Anderson via our website as well. Finally, I'd just like to give a quick reminder that we will be taking questions from the audience. You can submit them via email to hello@firstthought.io. Again, that's hello@firstthought.io. And thank you to all of those who submitted questions in advance of today's discussion. So with that, I'd like to turn things over to our moderator and veteran science journalist, Neil Canavan. Neil.
Hello. My name is Neil Canavan, and this is the second of 2 FirstThought webcasts focusing on the human microbiome and its impact on health. Specific to today, the microbiome's influence on the CNS. I came to know something about this topic in general in 2 rather different ways. The first is in writing my book, A Cure Within, published in 2017 by Cold Spring Harbor Press, where in the closing chapter is all about the nexus of the human microbiome and cancer. And as a self-serving aside, if you want me to send you a free signed copy, just send your snail mail to Morgen and she will send it to me and I will send it out. Now, second, I came to know about the microbiome as it relates to CNS because I've always had this stack of old journals and magazines on my desk. And periodically I raffle through them for ideas. And it was 4 or 5 years ago now, I pick up a copy of Scientific American from 2012. And this thing had a rather compelling cover on it. There it is. Now, this suggests that the we in us is mostly not us at all, but the microbial entities that call our bodies home. And the most compelling part of that related article was an interview with a man by the name of Dr. Sarkis Mazmanian. At the time of the reading, I was looking for speakers to do a keynote at JP Morgan healthcare conference, where my former employer was having an event. And I asked Sarkis to speak. He gave a talk on microbiome and the CNS, and I've been following his rather remarkable career ever since. And now to meet the man. Dr. Sarkis Mazmanian is the Louis and Nelly Soux Professor of Microbiology in the Division of Biology and Biological Engineering at the California Institute of Technology, or Caltech. In his career, Dr. Mazmanian has won numerous awards and honors, but really the sexiest one of all is the MacArthur Genius Award. This was bestowed on Dr. Mazmanian in 2012. Sarkis, welcome to the podcast.
Thank you, Neil, for having me. And thank you for refreshing my memory with that cover of the Scientific American magazine. I hadn't seen that in a while. It brought back the memories of that era of research for our laboratory.
All right, so let's go right to that. MacArthur Genius Award. If people online don't know what this is, look it up. It's pretty cool and a prestigious thing. I mean, people from the arts get them, from the sciences get them. Sarkis, real quick before we start in on this on our talk today, what was it like when you won? Were you surprised?
I didn't believe it. It's a little bit of a backstory. I'm not sure if we have time, but
sure.
They asked David Baltimore, the foundation asked David Baltimore, who's, as many of your audience will know, a Nobel laureate and a professor here at Caltech, to schedule a meeting with me. And he wouldn't tell me what it was about. The meeting started, it was actually the director of the foundation who took the call and not David. So they set me up, if you will, because they like to keep these things private. And so once I heard it, again, it came out of nowhere because I thought I was having a scientific discussion with David. And so I was speechless and really, like I said, in disbelief. And even to this day, I mean, it's an honor that many people deserve to win. I'm just fortunate enough to have.
Well, they chose well. Now, before we dive into the science, I want to give our listeners a bit of perspective. And for those that tuned into the previous talk with Dr. Jen Wargo, please forgive this bit of introductory repetition. Here's a few facts to get us started. There are roughly 100 trillion microbes in us, outnumbering our own cells 10 to 1, and they weigh cumulatively about 5 pounds. Second, of those trillions of bugs, roughly 10,000 distinct microbial species are represented in the human ecosystem. And every human is a host to a unique collection of over 1,000 of those species. Third, as for functionality, while the human genome includes roughly 22,000 genes that code for proteins, the microbiome has more than 8 million unique protein coding sequences, which means there are 360 times more active bacterial genes in you, in those microbes than in our bodies. So finally, what are all these bugs? Where are all these bugs? They're in your nose, your mouth, your ears, a lot of places. But mostly they're in your gut. As for what they're doing, principally they're helping us digest food, they produce vitamins, but relative to our talk today, they also have a pronounced impact on our immune system and potentially our very thoughts and behaviors. So Sarkis, I'm going to ask you to explain that last bit mechanistically in a few minutes, but I'd like to start with how you first became aware of this interplay between bugs and the immune system. The first paper I could track of yours related to that is 2006, the love-hate relationship between bacterial polysaccharides and the host immune system. Was that the beginning of your work?
It was, and by the time we wrote that review, we had already identified a number of different interactions between the microbiome and the immune system. And the genesis of this was that when I started my independent career, I wanted to think about areas of research that were different than the mainstream. And so my training was in microbial pathogenesis, essentially infectious disease, but the molecular basis of infectious disease. And I was working on Staph aureus and trying to understand different virulence factors, how Staph aureus causes staph infections. It's a noble endeavor, but I wanted to do something a little bit different with my career moving forward, and just started working on the microbiome, because again, we've known about these organisms for hundreds of years, the ones that live in our gastrointestinal tract and on our skin, as you mentioned, oral cavity, and elsewhere, other environmentally exposed surfaces. But very little research had been done on understanding beyond what the organisms were, but what are they doing and how are we tolerating them immunologically, and what benefits do we derive from them? You mentioned the production of vitamins and essential cofactors and minerals. A lot of that had been known for many decades, but I think applying modern tools, we were able to, maybe in the early 2000s, we were able to identify specific interactions, meaning that particular microbes and their products and how they affected immune response. And really the outcome, if I can just summarize, the outcome of that work was almost turning this relationship between microbes and the immune system that we had known or studied as a community for many years on its head, right? The idea here being, in these initial discoveries, that the immune system benefits from microbial colonization, that there are particular organisms that augment immune functions, that you make the immune system work better, if you will. There's a lot of details, nuance, specifics of that general comment that we can get into, but just demonstrating, at least in mice, initially in animals, more work being done in humans as the work has moved forward. But again, we show that there are specific T cell subsets that only develop in the presence of microbial colonization in mice. And again, we need these T cells for not just the immune and oncology applications that you mentioned in your CAR T series, but also obviously in fighting infections. And so again, really it's sort of this paradigm shift that the immune system essentially welcomes colonization by particular organisms so that it's more fit to fight off infectious disease.
Let's go. I pulled a cartoon from that paper. And so let's go a bit more into the actual mechanism involved here. I mean, I've been sitting in these lectures for 4 or 5 years now, and I still don't quite get it, how to control the Tregs and so on. But this is a pretty simple illustration. Walk us through what's happening here.
So we'll start from the top, and maybe follow the path of the microbe and its molecule as it impacts the role and the cellularity of the immune system. And so what you see from the top and what your audience sees is this organism called Bacterius fragilis, and this is in these brown symbols that look like bacteria. This is an organism that is found in roughly 50% of the healthy human population. And it's been studied in a number of different contexts, but what we were interested in was, again, how it interfaced with the immune system. And what you're seeing at the top in terms of the intestinal epithelial cells is that everything to the top in white of those epithelial cells is meant to represent the intestinal lumen, as you see. So the bacteria live in the gut, but again, they send their signals now. In fact, we know even beyond this diagram throughout the entire body. In this particular set of interactions, there was a specific molecule called PSA, or polysaccharide A, which is produced by Bacterius fragilis. It's a sugar molecule that coats the bacteria, and it's represented by this squiggly line with the positive and negative charges that's meant to represent its chemical structure. So the bacteria makes this molecule, PSA. PSA is sensed by dendritic cells, which are innate immune cells in the gut. They actually sample the lumen of the intestine. They're like sentinels, if you will, immune cells that are sort of probing that environment, looking for infectious agents, but also just trying to gauge what are the chemicals, what is the environment of the intestine. Mm-hmm. And so these dendritic cells internalize, they take up PSA. And what PSA then does is essentially reprograms the function of that dendritic cell. And what dendritic cells, like other antigen-presenting cells, do is that they synapse, or they interact with T cells and essentially both induce the activation of T cells and then fine-tune the function of those T cells. And what you're seeing in this diagram is that the dendritic cells that have taken up PSA, as I said, they take on a particular character, both at the genetic level, if you look at gene expression, but also at the functional level. For when these dendritic cells interact with T cells, they drive or promote the development of regulatory T cells, or Tregs, which mediate this balance between Th1 and Th2 responses. And so that's what you see on the far right, is that a balanced immune system, or a healthy immune system, has a proportion of Th1 and Th2 cells, but an imbalanced immune system, as you see on the left, may have more of these Th2 cells, which would skew the immune system more towards a particular allergic or pathogenic phenotype. So again, in essence, this microbe, via this specific metabolite, results in a homeostasis or balancing of the immune system, and allows the T cell repertoire to be positioned and ready to both fight off infection, I think something we'll talk about in a minute, but also to potentially prevent autoimmune reactions. Because the other aspect of immune function, besides control of pathogen, is that when the immune system is overactive or dysregulated, that leads to pathology, or what we would call autoimmune disease, or various different symptoms associated with that. So again, a balanced and really sort of adapted immune system is crucial for multiple aspects of health. Again, both fighting infections and preventing autoimmune reactions.
Okay, so I mean, there's obviously a lot to discuss here, and there's still much more to be discovered about this whole education of the immune system and how things can go wrong. But let's get to the genius part. I wanted to tell us about your epiphany, if that's what it was, that led you to dedicate your time and energy to investigating the microbiome in the CNS. The earliest paper I can track by you was PNAS. It's related to gut microbiota and the onset of MS, which is a neuromuscular disease with CNS lesions. But there's another article around this time, 2014, in the journal Neuroscience, and this seems closer to the mark. And there's a graphic here I want to bring up. This is nicely accessible. I really like this. It's easy to understand. but it is rather profound, and so much so I'm going to read the conclusion of that paper. Quote, these findings have resulted in speculation that alterations in the gut microbiome may play a pathophysiological role in human brain diseases, including autism spectrum disorder, anxiety, depression, and chronic pain. So would this paper mark the epiphany for you? And that aside, what about this work prompted that conclusion?
The way biomedical research works is by the time you've published a manuscript, the epiphany has happened already.
Yes, 2 years ago.
Generally, many years ago, many years prior to that. And so, but it is a sort of a depiction or a description of some of the early thoughts. And even 2014, as recent as that is one of the early stages of at least the recent study on the gut-brain connection and the role of the microbiome. So I have to caveat everything by saying that, most of what we've done, as a scientific community in this space, the groundwork was laid many decades ago, right? And I think that's true for many fields of research. And so I want to make sure we give credit to those early pioneers. We have just modern tools, modern perspectives, and resources to bring to bear. But that being said, some of the earliest papers on what we may loosely term the gut-brain connection were published in 2011, 2012, right? And we had already started working as a laboratory in this space. I'd say my personal epiphany was a conversation with a neuroscientist here at Caltech, Paul Patterson. I tell the story often. Paul was a neuroscientist and working on the environmental triggers for modulating behavior in mice. He had developed a mouse model based on epidemiologic data for risk factors for autism. He had just published that mouse model, and this is about 2008, 2009, when we were having this conversation. That mouse model, I can say to you now, is now widely used in the field, and Paul developed this. And long story short, in a very casual conversation, Paul mentioned that there were children with autism that had gastrointestinal symptoms, and he had just read very briefly about this. Again, this is well over a decade ago, and he didn't know much about it because, again, it wasn't his area of research. And so, again, it was just anecdotal data at the time, but based on that conversation, I told Paul, I said, I'm sure you're throwing the intestines of your mice away because you're a neuroscientist. And said, of course we are. What do we need with the intestines? And I said, well, why don't you when I send a graduate student over next time you have a group of mice, and on that first experiment where we looked at the intestines of those animals, again, this is now a mouse model that exhibits many of the behaviors associated with autism. Autism is a neurodevelopmental and behavioral disorder, but we saw many features of gastrointestinal dysfunction, microbiome shifts in those animals that exhibited these, autism-like behaviors. And so that was my epiphany, if you will, right? It's very rare that we see such striking results on that first experiment. Of course, it took many years to develop that into a scientific project, and one that where we were able to make discoveries. And it was in 2013 where we published our first paper, showing indeed that not only were there gastrointestinal symptoms in that mouse model of autism, but that one can intervene with a probiotic that both repairs the GI symptoms and improves many of the autism-like behaviors, not all, but many of the autism-like behaviors. And so it's a correlation. I mean, it's functional in terms of the fact that we're able to intervene with a specific human probiotic and improve both GI and brain function. Still trying to work on what those mechanisms are that connect the gut and the brain. I think this is an overarching topic, Neil, of thinking about how the gut and the brain communicate with each other. There's obviously a number of different ways that happens. And of course, it's important to remember bidirectionality. It's signals from the gut to the brain, and then signals from the brain to the gut that impact various different functions. But suffice it to say, again, to your point, after both that initial conversation and the set of experiments that were done subsequently to link that observation to a mechanistic phenotype, I believe that there was something here that could be investigated in mice, and then we've continued that research program. And in particular, now my laboratory, we've shifted away from the intestinal inflammation and largely the immunology focus of our research, and we're almost entirely now dedicating our research program to trying to understand how the microbiome affects neurodevelopment and neurodegeneration in mouse models.
We're going to get to the translational aspects in a few minutes, but first I want to talk just a bit more about the journey to get there. I know a lot of smart people, and on the face of it, there are some people that, let's say in 2014, would have said, reacted to this as like, you got to be kidding me. Was there a lot of pushback?
You know, I think that there's you know, there's different groups of people, right? So there's some people who, you know, who are this type of an interaction between the microbiome, and one thinks specifically about the gut-brain interaction, makes a lot of sense to them, right? So, you know, people who may have, you know, depression and they know that they have GI symptoms, or, you know, we've all, I think, viscerally felt a connection between our gut and the brain. I think one trivial example is, you know, if you were to either think or hear something that makes you anxious, you get butterflies, right? You know, so I think, you know, from that perspective, it makes sense. You know, from a more scientific perspective, I think there's just a lot that's unknown, right? And when one introduces a new perspective or a new topic, there's going to be healthy skepticism, right? I think that as a scientific community, we need that, right? We can't just believe every finding, because many of them, unfortunately, are either not reproducible or don't translate to humans, or, you know, are out of context. A number of reasons why, you know, sometimes, you know, a lot of sensational data isn't, that doesn't become validated over a long period of time, or again, it doesn't benefit humanity. But I would say this is a topic that I think fascinates a lot of people and really captivates a lot of people's attention.
Actually, let me go to that point as far as capturing attention. There's a rather profound conclusion you had here. Was there any interest from industry?
Yes, there has been. And the one point I just want to make is I also want to make sure that I'm not projecting just our own discoveries, our laboratory's discoveries. Again, there's a field of research now that's growing and emerging. So many other folks making really important contributions. And I think we may talk about that as well, Neil. Yeah. But you know, there has been and continues to be interest from industry, both in the microbiome space as well as in how the microbiome can be leveraged to access this conduit, this connection between the gut and the brain. You know, very early days, and you know, some technologies, some assets, even some, you know, very early stage clinical trials that have happened, in addition to sort of augmenting the preclinical mouse research, which obviously is easier to expand than the clinical work. But, you know, I think the best days are ahead of us. You know, I think we have some early indications that indeed gut-brain connections that we identify in mouse models are operative in humans. And, you know, what's really exciting about, again, we don't have, you know, we're far from therapies still, right? But just having that proof of concept that indeed we can intervene at the level of the gut and change behaviors in humans really opens up a lot of doors, right? And again, just one that's I think quite obvious to everyone is the current approaches to addressing psychiatric or neurodegenerative disorders are to try and get drugs into the brain, right? I mean, just generally speaking, right? But if one, you know, has a target in the gut that then mediates a beneficial effect in the brain, it's much easier, obviously, to get drugs into the gut and target those drugs to the gut without having them sort of get through the circulation. Oftentimes, they modified, some small fraction gets in the brain, you know, not in the right part of the brain, and there's obviously side effects. So I think this is the prospect, the concept of being able to drug the gut and get benefits for neurological conditions is one that really excites me and a lot of other people.
So let's stick with the example of autism for the moment. This describes like a wide variety and intensity of physical symptoms and behaviors. And I know gut GI issues are not uncommon to such individuals, but is a microbial related autism, is this a subtype, or do you find dysbiosis in all the patients that you've looked at?
Yeah, so it's unknown. The answer to your question is unknown. There may be particular subsets of individuals with autism where their microbiome is a larger contributor to symptoms, whether it's gastrointestinal symptoms, behavioral symptoms, hyperactivity, seizures, what have you, and others where the microbiome has less or no effect. But we don't have those answers yet, Neil. I think it's still early enough in the research. What we have are a lot of associations where changes in the microbiome or GI function in specific subsets of individuals with autism. But again, getting to whether or not those changes are contributing to any symptom or relevant outcome in humans or are a consequence of the disease state still needs to be worked out, right? So again, as powerful as these studies are in humans where we see microbiome shifts, let's say in certain individuals with autism, not all, but certain individuals with autism relative to match neurotypical controls, those changes could be because of their GI issues. It could be secondary, right, to GI. It could be secondary to their lifestyle, to diet, which is a major contributor to microbiome configuration. And so again, very powerful correlations, but experiments both in humans and in mice to show whether or not those microbes are indeed mediating relevant outcomes are underway. Again, as I mentioned, there's some early positive outcomes, but I don't think we're anywhere near knowing whether or not the microbiome is contributing to a subset of individuals with autism, and B, maybe a higher bar is, can we do something about that, right? Can we, target any sort of drug or interventional modality to, alter or improve relevant outcomes such as behavior, right? Again, others are very interested in this topic, but I do think, Neil, there's so much emphasis and so much growing emphasis because of this potential, I believe, because of getting drugs to the gut is something that makes sense to a lot of people, that I believe in the next few years we'll see more and more results and outcomes that suggest or not whether or not one can change behavior in a profound way by intervening through the microbiome. And so I think we're having this conversation at what is, I think, an important inflection point in the field.
So let's move a little closer to the translational work. This is, I'm going to talk about your most recent paper, or I think it is the most recent. This is from March of this year in the journal called Biological Psychiatry, which I did not even know was a thing. But you looked at bacterial metabolites. And the conclusion of this paper reads as follows. The findings support a connection between metabolism, gastrointestinal physiology, and complex behavioral traits that may advance discovery and development of molecular biomarkers for autism spectrum disorder. So this conclusion sort of begs the question that if you develop a panel of biomarkers, what's the utility of that? Do you use it in prevention? Does it give you a window to potentially treat a disorder? How would you use it?
I think potentially is the operative term, but both, right, would be avenues to pursue, both in terms of biomarkers and prospects for interventions. And so just maybe taking one step back, just generally, as I mentioned, the gut and the brain are in constant communication, and that happens through a number of different pathways. It could be, the immune system, the vagus nerve, it could be neurotransmitters that are produced in the gut and affect behavior. And one that we've focused on is the role of microbial metabolites, or small molecules produced by bacteria that may impact behavioral outcomes. And so well before we made some of our functional discoveries in mice and this human study that you referred to, there have been a number of papers in the field showing that metabolism is altered in individuals with autism, both in terms of systemic metabolism, and in some cases, people had shown that microbial metabolism was also altered, and which is consistent with the fact that microbiome is deviates or is different than in autistic individuals relative to neurotypical controls, because microbes produce molecules. Those molecules get into circulation, and in the studies I'm referring to, were detected in the urine. So this notion that there's an altered metabolism, including micrometabolism, was already established. What we show in this paper is, well, we expanded on that work, but we also showed that there were now fecal metabolites that were altered and distinguishably altered between neurotypicals and individuals with autism, and then identified many of those, our focus being on the microbiome, many of those that emanate from microbial metabolism or gut bacteria. And so the idea is that since autism is a behavioral disorder, it is diagnosed strictly based on behavioral metrics. That most individuals with autism, usually the earliest individuals with autism usually are diagnosed as about 2.5 years of age through 4 years of age. I think the mean is about 4 years of age for diagnosis. And it's known that behavioral interventions are oftentimes more effective when implemented early. And so, since there is no, blood test or scan or any other way of diagnosing autism, we still rely on behaviors. The issue becomes that a child must develop to a certain point where they exhibit certain complex behaviors, right? An infant doesn't have social communication behaviors the way a 2 or 3 year old does. And so, a child has to develop, to show behaviors before it's if they're deviating from what one would consider normal neurodevelopment. And being able to use molecular biomarkers, such as small molecule metabolites, to identify either at-risk individuals or maybe even more profoundly at-risk pregnancies, I think just potentially gives caregivers a head start on, watching out for symptoms and maybe, potentially now understanding the molecular basis, potentially even intervening at this level beyond behavioral therapy. So again, the gold standard for treating autism are behavioral interventions. But now, in addition to chemical fingerprints, which may, again, serve as biomarkers or ways of identifying a person's at risk, those same chemicals may be contributing to behavioral symptoms and then now allow you to either increase or decrease levels of specific microbial metabolites. Okay. So what I just described to you are sort of the outcomes of that work and what that work suggests may happen. But I'm happy to share some unpublished data with you to show that indeed, at least again in animal models and preliminarily in humans, that modulating microbial metabolites seems to be an avenue to impact complex behaviors associated with autism. And so again, maybe very briefly, is we've looked at so in that paper that you just described, that 2020 paper in Biological Psychiatry, which is a prominent journal in the autism field, by the way, we identified many dozens of metabolites that were altered, about 28 to 30 that were distinguishing between, significantly distinguishing between autism and neurotypicals. And then, the work to identify a function for one of those takes years. And so, we started working on one of those metabolites called 4-ethynyl sulfate. In fact, we started working on it a few years ago to try and get down to molecular mechanisms. Is this metabolite impacting behaviors directly? And if so, how is it doing so? So again, in a nutshell, again, this is unpublished, but we show that the metabolite is called 4-ethylphenol, which is produced from tyrosine by gut bacteria, gets into the circulation of mice. It becomes sulfated and then actually winds up in the brains of the animals. It affects the maturation of cells in the brain called oligodendrocytes. It prevents those oligodendrocytes from maturing. And when oligodendrocytes don't mature, they don't perform their function, which is myelinating neurons. Myelination of neurons is essentially like coating on a copper wire. It insulates the axons of neurons and allows electric signals to be transmitted more efficiently. So an animal that's exposed to 4-ethynyl sulfate, because its oligodendrocytes are less mature, because there's less myelination, just doesn't have the connectivity and the brain function of an animal that wasn't exposed to this metabolite, and manifests many anxiety-like behaviors and autism-like behaviors. So again, that's literally 6 years of work that I just described for one metabolite. We have many, many more to study, and obviously the combinations to study. So this is mouse research, obviously, so we take it with a grain of salt, but it shows that one metabolite has very profound biological functions at the behavioral level, at the tissue level, at the cellular level, at the molecular level in the brain, right? And so we now want to understand to what degree these phenotypes of this understanding that these metabolites aren't just a fingerprint, but maybe contributing to behavioral deficits in autism. We want to now understand those relationships in humans.
I wanted to come now to another part of your work. This is a bit more recent, and this has to do with Parkinson's. You published a paper in the journal Cell in 2016. This is called "The Gut Microbiota Regulate Motor Deficits and Neuroinflammation in a Model of Parkinson's." And again, I want to read this conclusion because you do have a bit of a flair for punchlines. Coupled with emerging research that has linked gut bacteria to disorders such as anxiety, depression, and autism, we propose the provocative hypothesis that certain neurologic conditions, which have classically been studied as disorders of the brain, may have etiologies in the gut. So I want to put the graphic from that we had before, but with the vagus nerve on it. And I want to recite a bit of the findings from the paper. The idea of the gut-brain axis stems from previous evidence that patients with neurological disorders experience GI problems earlier in the disease or at the time of diagnosis. 70% of neurons in the peripheral nervous system, meaning those not in the brain or spinal cord, reside in the intestines and connect to the central nervous system via the vagus nerve. If you could briefly tell me about the experiment that led you to this paradigm shift of an assertion.
Yeah, so as I mentioned a few minutes ago, oftentimes, the discoveries we make are rooted in literature that has existed for quite some time. In fact, in Parkinson's, to my knowledge, the first association between behavioral symptoms, which are tremors. In this particular case, there are obviously additional behavioral symptoms, so motor symptoms associated with Parkinson's, such as difficulty in walking or hunch posture. But the tremors were associated with gastrointestinal symptoms in 1817. Right, so an essay by James Parkinson called An Essay on Shaking Palsy, right, before it was called Parkinson, it was named after it. Parkinson's disease was later named after him. Dr. Parkinson talks about, again, 204 years ago, I think 6 individuals, 4 of which had GI symptoms. They all had shaking palsy. And then one of them, he gave a laxative to, and their shaking dissipated. And so this is, again, obviously, this is not new to anyone in the field, certainly not new to Parkinson's. The Parkinson's community is certainly not new to neurologists, that there are GI symptoms in individuals with Parkinson's. In fact, upwards of 80% of Parkinson's patients have gastrointestinal symptoms, namely constipation, oftentimes preceding motor symptoms by years, if not decades. Right? So again, this, people knew about this, but very little had been done. Surprisingly, very little had been done. I'm not sure I know exactly why. I've asked a few neurologist friends of mine of why people haven't pursued the GI aspects in terms of, not just treating them, but understanding to what degree does this contribute to the disease? Is it part of the pathophysiology of Parkinson's? Again, it's hard to know why that research hadn't sort of taken off, but oftentimes it's because, patients go see a neurologist who doesn't know how to treat their GI symptoms. They go see a GI specialist separately, and the two don't talk to each other, right? But we were emboldened, getting back to mouse research, we were emboldened by our discoveries in neurodevelopment, specifically in autism. And we said, well, here's a neurological condition, neurodegenerative disease, Parkinson's, that has this very profound gastrointestinal manifestation. Perhaps the microbiome is involved in Parkinson's disease as well. So very much a leap of faith, if you will, when we started the research, which culminated in a 2016 paper. Oftentimes, as I said, the work has been done many years prior to that. And so very talented postdoc at the time, now an assistant professor at Emory University, Tim Sampson, very intrepid individual, took on this project and showed that if he depleted the microbiome of a mouse model of Parkinson's with antibiotics, or if he rederived them in germ-free conditions, all the symptoms went away. right? So the mice behave normally, their neuropathology went away, their neuroinflammation went away. And so again, it's a binary result, right? So plus minus result if you have microbes or no microbes, but clearly, at least in that mouse model, it showed that the microbiome was involved in the exacerbation or the presence of the symptoms, and then went on to characterize specific organisms and their molecules which promoted motor symptoms. And then there's a line of research that we can discuss there. But while we were doing the work, the first paper in humans came out in 2015 showing that there's a microbiome change in individuals with Parkinson's. So again, we don't know cause or effect, right? Because these individuals have constipation, but their microbiomes were altered. So just in the past 5-6 years, this finding that people have known about for a couple of hundred years, it has now some biological or scientific rationale for it. Again, very, very early days. But we know, again, that the microbiome is not just different in humans and in mice that have symptoms of Parkinson's or Parkinson's patients, but that the microbiome, at least in animals, is contributing. So then the question is how, right? So getting back to what is that gut-brain connection? And then here again, we can leverage literature, some epidemiologic data showing that individuals who were vagotomized many decades ago are protected from Parkinson's. There's literature showing that individuals who have Crohn's disease or intestinal inflammation are at higher risk for Parkinson's disease, again, connecting the gut and the brain. And in mouse models, if you sever the vagus nerve, many of the symptoms of Parkinson's are ameliorated. And so again, there's no, nail isn't in the coffin yet here, Neil, but a lot of evidence suggesting that potentially there is a microbial trigger in the gut that can start a cascade of events that then lead to transmission of pathology from the gut up the vagus nerve into the brain, ultimately resulting in neurons in the brain dying and then leading to the motor symptoms of Parkinson's. It's a very convenient explanation for something called Brake's hypothesis. So Brake's hypothesis was proposed in 2003 that suggested that Parkinson's may begin in the gut or in the olfactory bulb. It was based on pathology of postmortem symptoms showing that the same underlying pathology in Parkinson's, by what I mean by pathology, are aggregates of a protein called alpha-synuclein. So it's a neuronal protein. When it clumps together, it's believed to cause a neuronal dysfunction, neuronal death. It was shown in 2003 that individuals with Parkinson's, many of them had the pathology in the gut as well as in the brain. So this led to speculation that potentially there's a GI involvement. So now if you now couple the more recent work in the past 5 or 6 years with the microbiome, again, it suggests that maybe the GI symptoms start early because there's neurodegeneration in the gut first, right? That's what's causing, gastric motility defects. And then over time, over many years, the pathology spreads. These clumps of alpha-synuclein aggregates spread through the vagus nerve into the brainstem and into the brain, potentially explaining why vagotomy prevents this from happening. Because if you cut the conduit, if you cut essentially the bridge between the gut and the brain for this particular pathology, the pathology doesn't reach the brain and doesn't result in motor symptoms, right? Again, a lot of work still remains, but I have to say that in the past few years, there's been a tremendous amount of support for this model. Again, many researchers beyond us, and again, a lot of enthusiasm because it gives us an opportunity to potentially intervene at the level of the gut for neurodegeneration.
Okay, we're going to throw it open for questions in about 5 minutes. But first, now that we've discovered the Houston, we have a problem question has been clearly answered. I want to talk for a few minutes about what to do about it. There's something called Axial Biotherapeutics. This is a company you have founded, and the work at Axial is trying to approach this problem from a drug perspective. So, what are you doing at Axial? Tell me what you're about your work.
And so Axial was started to essentially translate our preclinical, our basic research findings into people, and they've done an incredible job in doing so. And there's a number of programs at Axial, but the one that I think you're referring to, which has been released in the news, was a recent open label trial in autism. And so it's based squarely on the mechanisms of action that I just mentioned to you about these microbial metabolites. So I gave you this example in mice with 4-phenyl sulfate, which again is produced in the gut, gets in the brain, changes brain cell function, and ultimately induces behaviors consistent with autism. And so what Axial decided to do was that, once seeing this data, there were a number of different, ideas and ways of intervening, but Axial came up with this notion that potentially one beneficial strategy would be to keep these molecules in the gut, right? Because again, we don't produce these molecules that I'm talking about, which are in that biological psychiatry paper, the bacterial ones, and to the best of our knowledge, they're not produced by any organisms other than bacteria. And so if they're coming from the gut, the idea would be that if you can sequester them in the gut and keep them in the gut, they don't get into circulation, they don't get into the brain, and then cause these behavioral changes, these behavioral deficits. And so Axial developed a small molecule sequestrant, an oral, non-absorbable molecule that binds these small molecule metabolites, that binds these harmful bacterial products as it passes through the gastrointestinal system, and then ultimately the individual then excretes these metabolites with the drug, right? Again, effectively lowering circulatory concentrations. We tested in mice and it worked. It both lowered circulating levels of these metabolites and also improved behaviors that I had mentioned. And again, open label trials, so there's a placebo effect that's not accounted for, but the initial results are incredibly positive, that in 26 adolescents with ASD that completed the study, many of them improved in GI function as well as in various different aspects of behavior, most notably in anxiety and in irritability, which are associated with autism. And so again, early days, and we want to be careful about the message we send with this clinical trial. There's still a lot of work that needs to be done, but in terms of this first step, not only was a drug, I should mention, not only was the drug very well tolerated, very safe, but again, appeared to improve behavioral symptoms of autism in an adolescent autism population. So the next step would be obviously a randomized placebo-controlled trial, which is what the company will do. But again, the take-home message here is that with these orally available molecules, drugs essentially, we may be able to improve behavioral symptoms in autism. Again, not trying to overcome the challenge of getting drugs into the brain.
Okay, I'm going to throw it up for questions. Just 2 minutes, I promise, to the listeners. Just a couple more questions. I'm assuming there are larger trials in the works, and I'm also assuming there may be people on this call that have family members with autism. How does one get on one of your clinical trials at Axial?
Yeah, so they can contact the company. The trials are being performed in particular sites in particular countries. So there's a geographic issue. There's also, like any other trial, there are inclusion and exclusion criteria. So not everyone who even enrolls in the trial will be selected to participate in the trial. But they can contact the general. I think it's just info@axialtherapeutics.com, and we keep a repository, a list of individuals who have contacted us. But also there will be announcements going out as well for the upcoming trial. So people can keep an eye out on social media. But the initial phases of the trial will be in Australia and New Zealand. We'll expand into the US as the trial continues over the next year or so. So there, through social media, you should be able to find access. But again, I want to just, not raise too many expectations. Most of the people who contact us for the trial just simply don't qualify. Again, and that's true, as you know, Neil, for many clinical trials.
Okay, Morgan, we have a couple questions. Let's go ahead and start with those. For our listeners, we have 10 minutes left, so go ahead and dial in your questions.
Yes, and just as a reminder, you can submit questions via email to hello@firstthought.io. So one of our listeners, Dr. Mazmanian, had a question about the microbiome as it relates to the bloodstream. So they wanted to know whether, like, how does the microbiome contents get to the bloodstream? So do they pass through the intestinal cells, or do they pass between the intestinal cells?
Yeah, it depends on the molecule, it depends on the size of the molecule, the charge, the chemical structure of the molecule. The smaller the molecule is, obviously, the easier it can penetrate paracellularly, meaning between cells. And then the larger molecule it will have to cross the membrane, so there should be, particular transporters for different types of molecules. And again, the answer to the question is it depends on the type of molecules, right? So it isn't just one answer or it's not one size fits all. But suffice to say that many molecules or compounds from the gut do get into the bloodstream. Of course, we need this for digestion, for nutrition. And microbial molecules are no different. As far as epithelial transporters are concerned, they're going to transport certain classes of molecules. Our cells don't know the difference oftentimes between whether that molecule came from food or from microbes. And then but again, a lot of what happens in the gut does wind up in the bloodstream. And then from there, it can get anywhere, right? Any tissue in the body, including the brain. And then the other aspect to remember is that very surprisingly, right? This was kind of shocking to me at the time, but many neurological disorders have not just GI symptoms, but this phenotype called leaky gut, right? Or this barrier permeability that's been increased barrier permeability that's been measured. In other words, so our intestinal epithelial cells, which line our gut and keep the outside out and keep the inside protected, if you will. If that barrier, like let's say like a zipper, is opened, right, then things that aren't supposed to get crossed or supposed to be regulated as they enter now just sort of flood through the system, right? And so interesting, both autism, depression, schizophrenia, Alzheimer's, multiple sclerosis have all been associated with this leaky gut phenomenon. So that can also happen, right, where this discrimination breaks in the gut breaks down, and molecules, again, either class of molecules that weren't, normally transported, or their levels of ones that were now change in the bloodstream. So again, it's the molecule itself and the health of the host.
Thank you for that. I know we've covered a lot of CNS disorders, but there was a question from one of our audience members about whether or not there's been any research to a link between ALS and the microbiome.
There have been small studies looking at microbiome configuration in ALS, which is a neurodegenerative disease, and making associations and changes in the microbiome. And there are a lot of associations in the literature between microbiomes of whether it's disease, whether it's like different diets, different geographies, what have you. The microbiome is malleable, it changes. And a couple of mouse studies now, which show that indeed not only are the microbiome changes, but particular microbes and specifically particular molecules from microbes affect the pathophysiology of ALS in specific mouse models. But Morgen, I have to say, with ALS, there's literally a handful of papers at this point. So it's still very early. I wouldn't consider that body of literature, I would consider it very exploratory, potentially promising, but I'm not sure we know to what degree the microbiome at all or at all is involved in ALS. The body of work in Parkinson's is just much greater, and there's much more sort of tangible endpoints and credible experimental results that suggest the microbiome may be contributing to Parkinson's. But of course, the lack of evidence in ALS doesn't mean it isn't, but as of today, I just don't think there's enough to say that the microbiome is involved in ALS.
Okay, we have 5 minutes more if somebody still wants to send in their questions. We do have time, but for the moment, I get to do some more. Sarkis, you let us in on just how long it took to nail down a metabolite and what you might want to do about it. But we can kill bugs really easily. We know how to do that. Why not just do that?
You mean like antibiotics?
Yeah, phage.
Yeah,
I don't
so
know.
So, obviously this is one that's been considered by researchers in the biomedical community, and there are a number of different ways, right? So antibiotics is one, but one can think about removing organisms using different modalities. So I think the first, assuming that we've overcome the conceptual barrier in believing the microbiome is contributing to particular neurological outcomes or behavioral outcomes, and we've identified the organisms that are producing harmful compounds, let's say, right? Again, that's in that context. Then the question is, then how do you intervene? So long-term antibiotic use is one, but but there are drawbacks to this, right? There's no such thing as a designer antibiotic, which is species specific, that goes in like a laser and can eliminate one particular species or strain of organisms. Antibiotics, even the most narrow spectrum antibiotics, are pretty promiscuous. And so the issue there is that there's a lot of collateral damage to the microbiome, right? When you remove, larger groups of organisms, many of these organisms we need for health, right? And so antibiotics really haven't caught on. I'm not sure there are, I don't know of any actually companies that are developing antibiotic-based therapeutics to target the microbiome. There are newer technologies, whether they're sort of transposable elements that are meant to go in and delete particular genes. There's phage therapies that are being developed to remove specific organisms. But I have to say, this is not trivial, right? It's not trivial, both in terms of the precision, the specificity of these therapeutics, and the consequences to removing an organism from the microbiome. If you remove one organism, well, that organism is producing products that allow other organisms to thrive or to change their physiology, then you're going to lose that function as well. I think, if I may maybe want to take your comment one step further, I think we still need to get smarter about how these organisms are mediating their effects, and understand, not just find a bad bug, if you will, an organism that's harmful, we want to remove it, but try to understand, well, what is it about that organism that's harmful? Can we identify genes or pathways, products in that microbe that is mediating these negative effects, and then specifically target those? To me, that's a more tractable approach, and one that we and others are working on. But again, what this requires is deep understanding of biology, of mechanisms of action, right? Because that way what to target. Right. Well, and then I guess the other avenue would be like FMTs. Like, we don't know what's bad. We know something is bad. A person's microbiome is deviated, and so we can do a wholesale transplant, if you will. There are companies developing those technologies. I think they're crude, and they need refinement over the years. But that would be, in my mind, probably the leading way. That changes in the microbiome. These larger changes in the microbiome would happen in the near future.
All right, I have one more question about, from an industry perspective, if you will, and druggable targets. In oncology, you can't get the drug if you don't have the target. For the microbiome work, would you need to do an analysis of an individual's microbiome before you could reach for the powder in the bottle?
That's one way of doing it, and it happens often, right? You want to know what's in the microbiome, what's in a person's microbiome. But oftentimes, you started this webinar with statistics on the microbiome, all the genes that were there, right? So what oftentimes the issues we run into is that we may identify particular microbes or microbial pathways, which are, let's say, highly divergent between disease and health, right? Broadly speaking, right? But the problem is we don't know what the microbe, oftentimes the microbe hasn't been studied, its properties are not known. So you sort of run into this brick wall where you may have a target, but you don't know what to do with that target, right? And so I think that's where we just need to get a little bit smarter and understand, go beyond cataloging and characterization, which again, the field is doing, and get to mechanisms of action, functions for particular organisms or microbiomes. Then I think we can tailor therapies to give us the benefits we want without that, trade-off, without that collateral damage and harmful effects to the rest of the microbiome. So again, I still think that there's a bit of a ways to go before we have those very specific targets to drug.
All right, well, we're at the top of the hour. Sarkis, thank you so much for joining us today. Morgen, take us out.
Yes, thank you both, Neil and Sarkis, and to all those who listened this afternoon. Have a great rest of your day.
Thank you.
See you.